Hello, I'm Emily. I'm one of the technicians here in the microbiology lab. This is our specimen receiving area here. There are several things that we do here, but I'll show you a few of the containers that we use. This is one of our sterile containers. We get several different types of specimens in here, including urines, body fluids, tissues, and sputums. This is one of the main ones we see every day. If we look here, we have our blood culture containers. There's three different ones. We got aerobic, which is a green top, anaerobic, which is an orange top, and we have our pediatric for a lower volume. These go on machines in a separate area of the lab once they've been received. And over here we have our e-swabs. So this type of container is used for our nosocomials. This is what you get every time you would come into the hospital, such as MRSA, VRE, or ESBL, CPE. And we also get strep throats in this same kind of container, as well as some wounds and some Staph aureus swabs as well. So those are the main types of swabs that we use here.
I'm one of the MLAs here in the microbiology lab. We just received a tissue sample here, so I'm going to show you how we process that. First, I get all my supplies ready. We do a bunch of different plates for tissue because it's a sterile specimen. We have blood, chocolate, McConkey, CNA, and our two anaerobic plates, which is Brucella and Brucella Neo. So I've labeled them all, I've matched it with our patient specimen here, and I'm going to stack them all, and I'm going to set them aside. And I also have a thio broth here in my tissue grinder container. So what I'm going to do first, open my Petri dish. So I'm going to grab the tissue and put some in our Petri dish here. I'm going to open my tissue grinder and my broth, and I'm going to examine this tissue here, and I'm going to take a piece for my thio broth and a piece here for our tissue grinder. Once that's complete, I'm going to put the rest back into our CNS container. I'm going to close that up and set this aside. Going to carefully place tissue in the broth and tissue in our grinder here. No longer need this, so that goes in our biohazardous container. No longer need this. This goes in our sharps, and I set my Petri dish over there. We'd use saline with our tissue grinder, just like that. And I'm going to carefully take out the tissue grinder itself and place it in the conical tube here. I'm going to make sure it's nice and secure, and then I'm going to grind up my tissue. You can see that my tissue has now been grinded up. I now no longer need this. This will go in our biohazardous container. Now I'm going to grab a syringe. Put one drop on each plate. I put back, and I no longer need this. Last step is we do a gram stain. I'm going to write directly on the slide the specimen number, first two initials of their last name, and first two initials of their first name, and what test this is. I'm going to grab another pipette here, go back into our sample, grab my slide, place one drop on the slide, and spread it out. And then that goes on our slide warmer to dry. Close this all up. Invert our broth here to make sure our tissue sample's in there. And then our plates will then go on the isoplater to be struck and then incubated. And that's it. Thank you.
Hello everyone, my name is Raj and I'm one of the senior MLT working in the Biochemical lab, and also I do some teaching in the university adjunct assistant clinical professor at my master pathology and molecular medicine department. So today I'm going to show in the video how I'm going to extract the specimen that were already received in the lab, how they will be planted in the cultured media, and how they will be incubated overnight to grow bacteria if they have some in them. Let's see. So this is the sputum sample we received today in the lab. Since these are the clinical specimens, they might have so many nasty things. We need to process them inside the biological safety chemical as a safety precaution. That's what I'm going to do now. So you can see I do have 3 culture medium. That's what we usually do for sputum samples. One is chocolate agar, looks like chocolate to us. Another is blood agar, which has 5% C blood, and also I do have one McConkey agar, which is for selective growth of some of the bacteria. So to start inoculation, so I need to take a sterile loop. This is a sterile disposable loop. It has loop in the end. So I'm going to dip the sputum sample onto the loop to carry some of the samples supposed to carry the disease in question. So I'm now putting this sample on the one of the culture media, agar. So I'm going to do, this is the primary inoculum line. So I'm going to do primary inoculum line. This is how we're going to do. So from the primary inoculum line, I'm going to spray to the secondary. From secondary end line, I'm going to do the quaternary lines, and then finally I'm going to do the tertiary lines. So towards the end, that's the tertiary line. I'm expecting some of the isolated colonies, which is required for the further identification and susceptibility testing if they have some in there. So I'm repeating the same process over again with the second media, which is the blood agar. Same thing over again, primary line, going to the secondary line, going to the tertiary line, and quaternary line, aspirating some of the isolation. And then again, same thing on the third media, which is McConkey. So these are my plates set up. So after planting them or inoculating them in the agar, so these plates to go on at 37 degrees Celsius for overnight incubation. So now I'm bringing those plates over to 37 degrees Celsius incubator, where actually bacterial multiplication and growth occurs. So this is my incubator. This is how I'm going to leave them in the incubator. Thank you.
bottles at a time. So I already put 6. And now I'm gonna put the rest on the virtue too. And then it's just gonna take it and then distribute it to the 3 incubators by itself.
Hi, I'm Brittany. I'm an MLT at the microbiology lab at the Hamilton General. On this bench, we're doing blood culture grams. We receive blood culture bottles from our wards. We receive anaerobic and aerobic bottles. We then put them on our machines to incubate manually, or incubate on the machines. Our machines have different metrics where it can determine if it's positive or if they think that there's blood or bacteria in the blood culture. We then start a manual process where we run it on our WASP machine, or our walkway specimen processing. We have 3 samples running now. It'll set up our plates and our gram stain. We then, as techs, will stain our gram stain and read it and release the result to the ward. The plates are then incubated for about a day and then worked up. This is our Midas stainer. It is one of the best automatic stainers for gram stain. It most accurately mimics doing a hand gram stain. It uses dip protocol, so you don't have stain spraying on it. You tend to have a lot of over-decolorization when that happens. The Midas stainer is wonderful for staining bacteria of all sample types. We will stain blood cultures, spinal fluids, sputums, everything on here. This is what it looks like. It's a timed process. It's relatively quick. And it also does a methanol fixation. So most of these slides are fixed via heat and also via methanol, which can reduce any risk to the employees in the lab. It's now going into the water rinse. In here we have methanol, so that is our fixation reagent. In here is just running water to clean off the slide. This is safranin, so this is our final stain color. This is what stains our background pink. This is our decolorizer. So this is when you will see all the crystal violet iodine complex leave the cell wall of gram-negative bacteria. Iodine is our second stain that we use. Crystal violet is the first. So this is the most important factor in determining if an organism is gram-positive or gram-negative. For all bacteria, the crystal violet iodine complex will enter the cell wall. For gram-negative bacteria, the decolorizer will actually degrade part of the cell wall, the peptidoglycan layer, and that allows the crystal violet iodine complex to leave. So then these bacteria are just staining with our counter stain, safranin, which is pink.
Hello everyone, I'm going to show how to sort the specimen into the lab. So you have seen how we receive the specimen, how we process them, and how we incubate them into the incubator. After the overnight culture of them, we have to sort the specimen into the different area. So this lab is a huge lab, so we have many different areas and we work based on the area. Principally, we do have few significant benches. For example, anything from blood culture would go into blood culture benches. We have 2 of them. That's what we call the blood culture area. And then we have urine bench, we call the urine area. We have nosocomial bench where we do a lot of
surveillance specimen, for example, MRSA, DRE, ESBL, CP, etc., etc. So next one is the wound area. So we do have 2 sterile wound benches and 1 superficial wound benches. Anything from that area does go to that area. So also we do have a respiratory area where all the specimen from the respiratory specimen will go in that bench. Thank you.
So now I'm going to show how bacteria look like on the solid agar plates. To begin that, let me show you some of the agar. Broadly, these agar media, they are classified into there are different many ways of classification. The one very popular one is based on their performance. So they could be non-selective, which allows the growth of all the bacteria. For example, blood agar plate, I'm going to show you. This is the blood agar plate, which is non-selective agar, and also it does slightly differential function as well. As see shown in this picture in the plate, you can see a clear zone around the colony, which is called beta hemolysis. So basically this blood agar plate is non-selective media as well as partially differential media showing beta hemolytic reaction, which is the clear zone around the colony. That's the one category. And then in the meantime, it also shows you the alpha hemolytic pattern, which is another way to read hemolysis. In that case, you don't see the clear zone, but you could see the alpha or greenish zone around the colony. So this is the alpha hemolytic colony on blood agar. You can also see the alpha colony better on chocolate agar than blood agar. You can see the greenish discoloration around the colony better than chocolate agar. Although in chocolate agar there is no RBC to call hemolysis, but effect of hemolysis you could better see in chocolate agar. So that's one. Another one is differential agar. Let me show you the McConkey agar, which is a good example of differential media. So this has the indicator, which it gives the bacteria 2 different contrast color. The first one we call lactose fermenter. You can see dark pink colony on the agar, we call lactose fermenter, versus the other one which does not have color or colorless colony, which is non-lactose fermenter. So basically McConkey agar is differential media as well as selective media. So it stops or inhibits the growth of gram-positive bacteria, and that's selective function. And while it allows the growth of gram-negative bacteria, it gives you 2 different groups, growth groups, lactose fermenter and non-lactose fermenter. That's why this is also differential media. So those colony, now we have to do further workup, identification and susceptibility testing as required by our procedure. So there is another example of non-selective media. This is the very popular media these days. This is urine chromogenic agar. So basically urine chromogenic agar is non-selective media, but it performs differential function. You can see on those 2 plates, there is one dark rose, little rose colony. That's E. coli. That's how E. coli grows on chromogenic urine plate. Versus there's another group which is the darker blue colonies. That's the bacteria we call KES, Klebsiella, Enterobacter and Serratia group. Those 3 organisms would look like growing like deep blue, dark blue colonies and urine chromogenic agar. So urine chrome agar also functions like, in somehow McConkey agar to differentiate the growth of bacteria. So I'm going to show you few basic tests. The first one is catalase test, and then second one will be oxidase test. The catalase test is very popular in the routine lab. We do frequently every day to differentiate a group of bacteria called Staphylococcus from the Streptococcus. So if my test is catalase positive, I would think that's Staph group, Staphylococcus, and when it's catalase negative, that will be Streptococcus. So these are the group of bacteria. So I'm now bringing my identification into Staphylococcus group versus Streptococcus group by doing catalase test. So let me proceed to do catalase test. There are different ways to do catalase. I am doing a very simple method. It's called slide method. So I'm transferring the growth from the top of the colony onto my testing slide. So this is my Staphylococcus. This is my one of the test bacteria, and then I'm taking another one from another plate which looks a little different on the plate. So I'm going to differentiate which one of them is Staphylococcus versus which one is Streptococcus. So these are my two test bacteria. So now it uses 3% S2O2 hydrogen peroxide solution. So I need to drop on the colony. That's what I'm going to do now. This is ready to use media reagents, stable at room temperature. I'm dropping on the top of the colony in the slide. You can see the bubbles coming from the colony indicating catalase positive reaction. So what is happening in here is bacteria is producing catalase enzyme, which is breaking down hydrogen peroxide solution into oxygen, and you can see the evidence of oxygen bubble on the slide. That one was the positive reaction. Moving on to second one, my second test bacteria, I did the same thing on that, and you do not see any active bubbling like before. So now interpretation is the one which did give us active bubbling immediately after dropping the solution is catalase positive, possibly Staphylococcus group, versus the other one, second one, which did not do any bubbling is Streptococcus, and so it is helping me to bring my identification onto Staphylococcus and Streptococcus respectively. So the next one, another biochemical test is the oxidase test. I'm going to do now. So oxidase test is basically done again to differentiate the group of bacteria. The most common one is Pseudomonas aeruginosa from other enteric bacteria which belongs to the same family. So however, oxidase positive reaction is shown by other bacteria as well other than Pseudomonas aeruginosa. For example, Aeromonas, Vibrio, Plesiomonas, Neisseria, all of them, they produce the oxidase enzyme like Pseudomonas. So to do that, so you need a oxidase reagent, which I'm going to do on again filter paper on the slide. This is the filter paper method and slide method. So what I did before was, so I took one filter paper strip and I put on the slide, on the surface of the slide, and I drop a solution of oxidase reagent onto this is the oxidase reagent onto my filter paper. I'm allowing it to little dry so that I could see my results easily. So this is my test strip ready for oxidase testing. So the next step would be I need to transfer my test organism onto the oxidase reagent strip, which I'm going to do now. So this is the Pseudomonas aeruginosa growing on the blood agar plate. I'm doing that now. I'm transferring some of the colony and then smearing over the oxidase reagent. Just you look for change in the color. So oxidase positive organism will produce changes the color, develop the purple color immediately within 10 seconds. You have seen that with the Pseudomonas aeruginosa. This is oxidase positive reaction produced by Pseudomonas aeruginosa. Now I'm going to show another one which is oxidase potentially negative. Say this is my E. coli from blood agar plate. I'm again transferring my colony, and I'm doing the same thing on the oxidase reagent strip. I'm smearing on the filter paper with the oxidase reagent, and there you could see no change in the color indicating negative reaction. The one with intense purple color within 10 seconds indicating oxidase positive, which was Pseudomonas or Pseudomonas group having oxidase enzyme, versus the other one is I did was E. coli, which is oxidase negative reagents, but it's still oxidase negative organism belong to the same Enterobacteriaceae group. I'm showing one of the common bacterial identification process in the lab. We call it MALDI-TOF. To do that, so we need a MALDI-TOF target slide. These slides comes from the vendor. We have to buy them. Total 48 spots in one slide. That means you can do 48 isolates, 48 patients, whatever, in one slide. So you can do 16 group of 16 at one time. And this has 3 groups. Now I'm gonna do the third group. The first and second one used to be already used up. So I'm gonna do the third one. So to do that, first you need to do the control spot of E. coli. So I'm gonna do the control spot. So this is my E. coli, and then I'm gonna smear on my control area for the third group of 16 spots. This is the control. So after control, I have to do my patients. So I'm doing my first patient now. Just touching the side of the colony. Usually in microbiology, you have to touch 3 to 5 colonies. I'm just touching just side of the 3 colonies, 3 to 5 colonies, and then smearing on the target slide. So this is my eye. I'm gonna do the hair. This is the spot, row I, and then first spot. This is the one bacteria I like to do identification during MALDI-TOF. Let me do second one as well. I'm gonna repeat same process over again, trying to take representative colony from 3 to 5 isolated colony, make a thin spot on the target side. It's really tricky mass spec by this method. The success of MALDI depends upon your smear quality. The thinner the smear you make it, the better the chance of getting identification. So far I have put my bacterial smear on the target slide. So after that, since this is MALDI-TOF, it involves the use of matrix. I'm putting matrix solution onto those spots, which helps to release the bacterial cell, to release the DNA content, the protein content of the cells. So this is my control, E. coli control. I just drop 1 microliter matrix on that, and I have to repeat same process for other patient sample as well. This is the second one, and then another is the third one. So we have to wait until the smear air dries up. When it is dry, we can load this slide onto the MALDI-TOF instrument, which we're gonna show after this. Now let's go to MALDI-TOF instrument and see how does it look like and how it functions. MALDI-TOF instrument that I'm going to load my slide on. This is the next generation MALDI system, which has really great throughput for us. We can do 16 slides like this at a time go down. So this is the loading dock. We can load the slide right on here, and slide will be taken inside into the acquisition chamber. So it's going in right now, and then when it's really into the system, and then we can see it does acquisition. We call the acquisition. So in this case, so what happens in here is individual those spot that I showed you. So will be there is the laser on the top of the column. It goes in the column where there is a laser, and because of the laser, the bacterial molecules will fly in the vacuum, and based on the height or travel of the molecules in the air in the closed vacuum, it gives you the identification. This is the anaerobic bacteriology section in this lab. We call it anaerobic chamber. We play a 55 anaerobic workstation. What happens is some of the culture we do in the lab, we need to do anaerobic culture as well, especially the body fluids, absolutely sterile specimen. Those would get dedicated anaerobic plates set up. Those plates would be into this chamber for 5 days. We read them every day. If there is any growth, then we have to do anaerobic identification. You can see plenty of plates inside. So because we keep them for 5 days, they are organized by the day. Day 1, day 2, day 3, day 4, and day 5, and the text would come and we have to play every day 5. So in order to check if this has actually anaerobic condition maintained, we do some of the QC stuff every day to satisfy it's keeping all things anaerobically.
You can see in there some of the QC plates as well as the anaerobic QC strip, which has turned white, so indicating that's keeping anaerobic condition. So on the day one, we would load the plates on one side and this side. So this is the access door. We can load the slide. We can load our plates inside there, which keeps anaerobic. And once it is in, we have to move them into the body where there is actual 37 degrees Celsius. So this whole chamber, except those two doors, has 36 degrees Celsius. So we move the plate inside. We incubate them for 24 hours. Read them. Call in the process, keep another 24 hours, read them until 5 days, and in the day 5, we read them we make the final reading and then throw the plate as needed. So in this video, we are showing how Biotech 2 produce antibiotic susceptibility testing in this lab. So Biotech 2 is the only way we do susceptibility for most of the part of the lab. However, we do have some other methods as well. For example, Kirby-Bar. So in Biotech 2, what we do is we have to make the suspension of the bacteria to achieve 0.5 McFarlane standard, which has expected number of bacteria in that suspension. So that suspension would go into the Biotech 2 system, which would further dilutes using the sterile saline within the system, and those diluted culture suspension would be sprayed on those individual wells. We can see in here now there are you can see the individual wells. So Biotech 2 system inside the instrument will dispense that suspension onto the individual wells. So in those wells, it will be incubated 37 again, and then it reads every certain interval. For example, in this case, every 15 minutes. That's how Biotech reads. So if there is any change in optical density, it will record that. So what is happening in here is you can see in close view, you can see some of the wells, like micro wells. You can see the growth, turbidity indicating the growth, versus some of them you can see the clear. So the turbid well means that's not the MIC, minimum inhibitory concentration of the antibiotic, that indicating there is some resistance, versus well next to the growth well is whenever there is clear well, that indicates that's the MIC, indicating bacteria are sensitive to that particular antibiotics. That's how Biotech 2 will calculate which antibiotic is sensitive versus which antibiotic is resistant for individual antibiotics, which is in there already in the micro titration wells. Let's go in the next slide and see how does Biotech 2 instrument look like. These are the Biotech 2 instruments we have. We have 3 of them in the line. This is the smaller version, and there are 2 bigger versions with the full capacity. Each instrument can do up to 120 tests at any given time. So what we do is, I was showing you, so when we receive this suspension like that, so these are the suspensions for Biotech 2. We're gonna put those suspensions onto the Biotech 2 system. The suspension will be loaded in here. This is where the loading dock is. It is taken inside. And as already described, will be further diluted and inoculated onto those individual wells. So they do have incubator. So they do have incubator inside that keeps 37 degrees Celsius, as well as they have the reading station we call optical station, where the reading of optical change occurs. So as we mentioned, there are other methods of doing antibiotic susceptibility testing other than Biotech 2. I'm going to show you a few of them here. These are the alternative ones for Biotech 2. So the first one is Dix diffusion testing, we call Kirby-Bar. So we will use Miller-Hinton. The name of the media is Miller-Hinton agar. You can see that. This is very specialized and definable culture medium we use in the lab. What is happening in here is we have to make the inoculum, we call carpet culture or lawn culture. The suspension 0.5 McFarlane will be evenly spread in the agar.
using the sterile swab on the day 1, and then we have to apply the specific antibiotics you are testing on the agar. You can see one of them in there, which is the ceftriaxone antibiotic testing against one of the E. coli bacteria. And what happens is we have to incubate this again, plate at 37 degrees Celsius for 16 hours. While doing that, what is happening is the antibiotic will be diffusing away from the agar where it has originally spotted. That's the diffusion of antibiotics. And since the agar contains the bacteria everywhere, like lawn culture or wherever there is interception of bacterial growth versus the antibiotic, you can see the clear zone we call zone of inhibition. We have to measure the diameter of the zone of inhibition, and we do have interpretation guideline based on CLSI to interpret the zone of inhibition. So we could interpret as sensitive or intermediate as resistant based on the CLI recommendation for the zone of inhibition against particular antibiotics. We do have guideline in the lab for that. So this is another dish diffusion. But this has been done a little differently. You can see many antibiotics, 5 antibiotics on the same plate. This is the ESBL, we call as extended spectrum beta lactamase confirmation by dish diffusion method. You can see some of the antibiotics. One of them does not have any growth, has growth all the way, that is resistant, and there are others which has there is a distant zone of inhibition. So again, we do have to measure the zone of inhibition against each antibiotic and interpret accordingly. So this is the dish diffusion by Curvivar, and another one is the E-test. This is the blended method of dish diffusion and MIC, that is the minimum inhibitory concentration method. You can see it's the same principle. You're gonna make your lawn culture on the plate from 5 McFarlane growth. Instead of applying the one antibiotic which has one breakpoint, in this case we have applied a strip of antibiotics which has a gradient of concentration of antibiotics from very low, for example in the case 0.016 microgram per liter to 256 microgram per liter. So it's giving you the MIC, and how to interpret this one based on the intersection of the bacterial growth versus where it meets the antibiotic strips. So in this case, I can see the growth up to 1.5 and not at 2 MIC. So I'm going to read this as 1.5 MIC for the vancomycin strip against the Staphylococcus aureus.